Our last speaker for today's part of the showcase is Dr. Jonathan Ryves from Cupid-Peptides. Dr. Jonathan Ryves is the director of the Cupid-Peptide Company. He graduated from Edinburgh and gained his PhD in biochemistry from the University College of London. He has over 25 years' experience in protein biochemistry and molecular biology, working in UCL and Cardiff University, leading to over 40 peer-reviewed scientific publications. He first published in the field of cell-permeating peptides in 2006, and realizing the power of this technology firsthand, has focused on developing its practical applications since then. And today he's going to tell us about Cupid's technology for carrying large molecules into the cytosol of living cells with a novel cell-penetrating peptide.
Thank you very much. It's a real pleasure to be here, coming over from over the pond. Beautiful weather, beautiful place. So I have founded this company in Cardiff, UK, to look at cell-penetrating peptides anew and basically make a new platform for use, both in research and medical kind of use later down the line. So, Cupid technology, the relevance of CPPs for research, we think has been really underestimated. And the new generation of CPPs is powerful and versatile and easy to use. And they open the door to revolutions in many biomedical and therapeutic areas, and we want to be at the forefront of that revolution. So when we talk about CPPs, they're often classified just about their chemistry, whether they're basic, whether they're hydrophobic, hydrophilic, but we kind of want to look at it from a functional point of view, a biochemistry and a biology point of view. So with that in mind, we've made three classes. The first class is synthetic peptides, which is essentially polyarginine, polylysine, which you're probably aware of. They are positively charged, and they stick to the cell membrane, and then through the natural process of membrane recycling, they're taken into the cell. Similar to them, class 2 viral peptides, they are more evolutionary sort of based in that they belong to viruses that want to target cells and stick often to proteoglycans on the surface. Again, they have, once they've stuck, they get taken into the cell through endocytosis. The third class is one that Cupid belongs to, which is, again, a small peptide, 16 amino acids, is an amphipathic molecule. It adheres to the outer membrane through charge, certainly, but then our evidence suggests it passed directly through the bilayer and goes into the cytosol, where it can really do its work. So the problem was revealed by Lundberg in an early paper, early this century, if you like, and that group looked at class 1 and 2 CPPs, and what they found was when you look at them live, this is just in 5 minutes, and they've attached each one to GFP, so they're quite large molecules now, 30,000 kD. You get this characteristic round the membrane situation with these class 1 and 2s. And when you fix them, however, it looks as if the peptide is everywhere. Worse than that, if you like, if you actually fix the cells first, then add your peptide, you get this pattern. So it causes a real problem in understanding what's going on and interpreting results. When you look at the live cells, they found that, again, they're going around the periphery, attaching to the cell. And even 24 hours later, they're getting into the cell, but in a punctated form. they're basically trapped in vesicles, most of the protein. And in fact, large parts of the cell are excluded from the CPP. So anything it's carrying will not get necessarily where you want it to go in this situation. So just to sort of summarize that, both these classes of CPP are getting into the cell. They're being sequestered in vesicles of whatever form. And the issue is, how can they get out? Some certainly do, if they're very potent. There's lots of papers that suggest activity. But the fundamental problem is, are they actually going where you want to go? And if you've got to add a lot, are they actually going to be kind of gumming up the cell? So we developed along these lines Cupid attached to GFP. So there's our construct, just our tag to isolate it, Cupid, and then the GFP protein. Now, when we purified it, we denatured it, so it is not fluorescent, and that's very critical in what I'm going to show you now. So when we add this to live cells, and this was done by Dr. Chris George, National Heart Institute, first thing to notice is that, if you like, time zero, there is no fluorescence. The product you added is not fluorescent. Then, as time goes by, and we took 5-minute pictures on living cells, and you don't need to wash off during this experiment, so again, it's very easy to use. The fluorescence builds up and up and up, and when you analyze that data, you've got this sort of sigmoidal curve, probably reflecting the kinetics of GFP refolding through fluorescence within the cell. And the time span is pretty quick, 1 hour, so you've got a lot of capability of doing fast research with this sort of material. So looking at these cells now, the same cells we saw in the last one at 60 minutes, just confocal, we can see that it's present throughout the cell, each cell, and there's no holes where the nucleus is, and nothing we can really say is not being reached, if you like, by the fluorescence. Similarly, we never saw it in punctate form or even on the membrane during this experiment. It works in human cells. This is much lower dose, 1 micromolar. Again, non-fluorescent. Live human hex cells, no wash-off. And this is just a confocal section at 1 hour, again, showing it's present all the way through the cells. And this was done with the rather nice microscopes with Dr. Lewis Francis in Swansea. It didn't appear to be toxic at the levels we use, and the cells showed normal viability with the MTT test. So really, our criteria are being satisfied by Cupid-GFP. We have a product, we can purify it, it's pure, water-soluble, stable in storage. It can carry large cargoes, 30,000 is decent size. It's not toxic. And it's able to directly access the cytosol where the cell refolds it. And subsequent, if you have something attached to it that isn't GFP, that would allow target interaction. And traction of being initially non-fluorescent allows us to track it throughout the experiment. So a couple of examples of CPP3, the third class that I've personally been involved in, is one with amoeba. So when these amoeba starve, they live singly, eating bacteria, quite happy. No bacteria left, they starve. They all rush together into an aggregate. So that's what would happen when they starve. No treatment, they aggregate. And they do this through one essential action of protein kinase A. And there's a little peptide that inhibits protein kinase A, PKI. So this is an experiment with PKI. And we used PKI because no small molecule could get into the dictyostelium. They're quite rugged creatures. So we wanted to find a peptide that could do the job. Peptide alone doesn't do the job. Cupid alone doesn't do the job. But when you add them together, you show that it's inhibiting PKA. So basically the the individual amoeba go, I'm starving, where is everybody? They can't find each other, and they fail to aggregate. This was the second sort of example I'll use. There's these two proteins, which Dr. Britta Eickholt was looking at in neurons. And it's the P10 protein and the drebrin protein. And through a very elegant way, she labeled both of them with fluorophore, which then could connect with FRET. She could find out whether they were bound together or not, just through the FRET interference pattern. And she wanted to prove that the area where they connected, which was called the D loop, she wanted to prove that that was critical in their interfering with each other, I mean, in binding together. So she said, okay, let's make a CPP3 D loop protein. And the essential point of this slide is when you add the protein, it stops them interacting. So whether you add the stimulus or not, you are not having these proteins together. So I've been making a lot of different products. And this is the idea of protein mapping. So you get your protein, which is the big blue bar at the top of each picture. And then you can take individual pieces out, attach them to Cupid, and you've got a whole series of products, each of which should cause a different effect according to what the literature says that each part of the protein is doing. So you can kind of map the protein. So you could map a protein you had no idea what it did just by kind of looking at it and taking sections, attaching Cupid, and you've got a new product to test. So this sort of method, really we've been honing down to get it from design to peptide in your hand in 2 months. And we've been working with academics in Cardiff with a lot of projects starting here. So here's one of the prototype I call it, because it's only been done in the last few months. So this is the idea of the biggest we've done so far. This is Cupid with GFP and KLF4, which is a great one of Yanamoka's stem cell factors. So very interested in what peptides could do in that area. As you see, it's huge. I mean, 83 kilodaltons, and there we're finding it getting into cells under the same conditions I previously shown. So we're very confident that as a basis for research in many different areas, Cupid is a good platform to start with. So it's representing a new disruptive technology for protein research, for therapies. It's early stage development, of course, and pursuing it now offers the opportunity to gain substantial advantage over your competitors. So I recommend it to you. Thank you very much for listening.